Another demonstrator, cellulose acetate membranes for hormone removal from drinking water. The aims in this case is removal of steroidal hormones and improvement of Folin properties. As an optional aim, there is also introduction of antimicrobial properties. Our approach in general is activation of surface. It means deposition of some layers on the surface, then immobilization of nanoparticles, which are in the next step derivatized or covered by some receptors which are responsible for hormone removal from water. One of the production routes to produce nanoparticles is doping of hydrophobic matrix with cellulose acetate and mixing with hydrophilic modifier, and it was shown that we prepare nanoparticles which are having hydrophobic core and hydrophilic surface, which can be then used for the membrane functionalization. What was achieved with this demonstrator? There were stable dispersions prepared, and these were resistant against mechanical stress, which is very important for the membrane separations. There was successful immobilization of receptors achieved for this hormone removal issue, and we prepared highly active coatings which were resistant to standard cleaning procedures, and currently also removable approach worked. It means that the coatings stay stable towards cleaning procedures, but in some conditions we can remove them and regenerate them, so the lifetime of the membranes can be enhanced by such approach. Coming to the third demonstrator, the current state is there are no antimicrobial modal yarns or fibers available on the market. As you can see on the picture, this approach that we applied here was quite successful, because the T-shirts that you can see are outcome of the first production already, and there was approach applied where the silver nanoparticles were immobilized in soldier matrix. Another approach to achieve this is ongoing. There is in situ synthesis of silver nanoparticles running at the moment, and utilization is a variety of fields like medical applications, underwear, military or sportswear, textiles. A very interesting site and approach to transfer academic knowledge into industries, lifecycle assessment. And here I show illustration for paper, while the whole system of production is analyzed by lifecycle assessment, and applying some improvements. Either the process as a whole is analyzed, which you can see on the number 1 side of picture, or also some savings of the material and impact on the environment are analyzed, which you can see on the second approach. And here are some results which can be shown. You can see basically comparison of energy use, either renewable or non-renewable energy, and glasshouse gas production for different processes. On the left side, in the blue color, you can see conventional paper production, which is based on virgin pulp, and comparison what happens when the nanoparticles are inserted into this production. On the right side, you can see analysis of the whole process, and when virgin pulp is partially replaced by recovered pulp, there can be savings achieved, quite reasonable savings in energy costs, and also slightly less production of glasshouse gases. As another illustration of this approach, there were costs of preparation of different chemicals, nanoparticles analyzed. And as you can see in bold, using noble nanoparticles like gold, palladium, platinum, and rhodium is in a higher amount kind of nonsense. And therefore, even for silver, one has to take into care that the amounts should be really on nano or very small, nanomolar or small level. And the last part are the scientific hot topics, kind of side chain, not connected directly with demonstrators. So as I mentioned already, there were some demonstrators where it was important to achieve antimicrobial activity. And analyzing current approaches toward testing this antimicrobial activity, there are only plating out techniques available at the moment. And my colleagues found that these techniques are subjective, they have quite bad reproducibility, they are time-consuming and require really scratching of the cells and transfer them for counting under microscope. There was an approach developed within our project which combines scan electron microscopy, which is optional step at the moment and will be removed later, fluorescence staining, where dead and live cells are visualized, and real-time PCR to count or quantify the bacteria directly on the surface. So here you can see schematic visualization how the method looks like. So basically, sterile modal surfaces are fixed onto cultural well plates, contacted or incubated with Escherichia coli strains, and then their SEM measurements, fluorescent microscopy, and real-time PCR are run. I will show some results to give some overview. On this picture, you can see basically untreated cellulose acetate, which is material of production of separation membranes at the moment, and it is well known that this material suffers from microbial contamination when staying a long time. So you can see that there is lots of bacteria growing on this surface. Under this picture, there is chitosan-coated cellulose acetate. While knowing that chitosan has antimicrobial activity, you can see there are just few cells on the surface, and you can see these cells are dead. The cytoplasm is visible, I hope you can see it. To confirm this, fluorescence staining was made, and this was made just for chitosan-coated cellulose acetate. And this method works so that there are two dyes applied to the same sample, while one can penetrate to the membranes while other cannot. So you can see, as the pictures are showing the same color, the cells are dead already as the membranes are not existing anymore. And the third picture shows how much cells are present at cellulose acetate surfaces which are non-coated and on the coated surfaces, while confirming that these cells are already dead. Another activity that is running at the University of Graz is molecular modeling of cellulose water interfaces and absorption kinetics. Basically, there is at the top list of programs which are used to do this. I'm not going to come into details. I would focus on the pictures on the right side, where you can see results of some modelings, which sometimes take several days. And this structure of cellulose 1 alpha and cellulose 1 beta structure, while for cellulose 1 alpha you can see rough character of this structure, which results from tilted cellulose molecules, and cellulose 1 beta is a really smooth surface, and there are water molecules interacting with these surfaces. To summarize achievements with these activities, there are different molecular models made of different cellulose allomorph structures, as I showed in the previous slide. There were interactions modeled, and position of water molecules was calculated. There are some studies on interaction with small saccharide molecules ongoing, and as driving forces, Coulomb and van der Waals forces were calculated. Coming to the last hot topic, which is also running between University of Maribor and Uni Graz, is development of microarray sensors for surfaces, and it was successful to use this attitude for the specific absorption of DNA. Basically, the aim was to avoid non-selective absorption of protein DNA, which was reduced to a high degree by our approach. Then immobilization of high amount of receptors was achieved. The method was used was based on the fluorescence light which was produced by hairpin effect, and this method was shown to be highly selective and can discriminate between 20 and 19 complementary nucleotides in DNA chain. So this was all from my side. I would like to thank you. This is my team in the first product of SURFUNCELL project, the Antimicrobial Tshirts. Thank you.
Thank you very much, Dr. Tatarova, for this interesting overview of the project. If there are any questions, we have time for one question. Please go on.
The silver nanoparticles on textile, did you try if they are washed
course, because there are strict limits to achieve antimicrobial activity. Of course, with the first attitude that I showed, which is in production, there is some washout, not that much, but after 20 washing cycles, there is still antimicrobial activity achieved. And at the moment, it's quite fresh and it's in the lab. At the moment, there was attitude achieved where the washout is almost zero. So we are trying to put it into production scale.
Okay, thank you very much. If it is a very quick question, go on.
It's a quick question. You listed a long, impressive list of your achievements. I was a bit concerned that you see nothing negative. And
it's not true.
For example, concerns with health hazards, which are quite a big concern, especially for nanocomposite, etc. Don't you think it's also an important issue to treat?
Yeah, I think you are right. But the time of 20 minutes is limited, and one would like to really list the achievements of the project better than attitudes that were not working. But I must honestly say that the students working on such projects, which are focused on some pilot experiments and production, really have to work really quickly, not to really think about why some attitude is not working, analyze it into details, and coming with some new ideas and really trying to find something that is working. That's different kind of attitude with such projects.
Okay, thank you very much. We go ahead with the next speaker. So we have... Thank you.